Have you ever wondered how a cell knows when to produce a specific protein and when not to? This fascinating process falls under the realm of gene regulation. It's an intricate dance of cellular function, ensuring each protein is produced at just the right time. An intriguing example of this is the Lac Operon, a system within bacteria that regulates the production of lactose-digesting enzymes. So, how does this all come together? To understand how this works, we delve into the world of the Lac Operon. The Lac Operon, a fascinating system, exists in bacteria like E. coli. Now, you may be wondering, what exactly is this Lac Operon? Well, it's a set of genes, specifically 3, that work together to control lactose metabolism in the bacterium. These genes are neatly organized into a unit that includes a promoter, an operator, and the genes themselves. The promoter is where the RNA polymerase binds to initiate transcription. The operator is a DNA segment that acts like a gatekeeper, and the genes encode proteins for lactose metabolism. This elegant setup is a classic model system for understanding gene regulation. It's like a finely tuned orchestra, with each component playing its part to produce a harmonious outcome. In this case, the efficient use of lactose as an energy source. So the Lac Operon is a switch, turning on or off to control lactose metabolism. But how does it know when to switch? The Lac Operon's magic lies in its ability to sense the presence or absence of lactose. Now, let's delve into its fascinating mechanism. Imagine the Lac Operon as a switchboard. The repressor protein is the key player here. It's a bit like a guardian, keeping an eye on the operator, a part of the Lac Operon. When there's no lactose around, the repressor protein binds to the operator. This binding prevents the machinery of the cell, known as RNA polymerase, from transcribing the genes downstream of the operator. In essence, the repressor acts as a roadblock, preventing the production of enzymes that break down lactose. But what happens when lactose enters the scene? This is where the real magic happens. Lactose, or more specifically, a derivative of lactose called allolactose, acts as an inducer. It binds to the repressor protein, causing a change in its shape. This shape change inhibits the repressor's ability to bind to the operator. With the repressor no longer blocking the way, RNA polymerase can now proceed with transcribing the genes. This transcription results in the production of enzymes that can break down lactose into simpler sugars, which the bacteria can then use for energy. What we're seeing here is a classic example of gene regulation. The Lac Operon is essentially a system that can turn genes on and off in response to environmental cues. In this case, the presence of lactose triggers the system to switch on the genes that help the bacteria utilize lactose for energy. This regulation ensures that the cell doesn't waste energy making enzymes when there's no lactose to break down. But when lactose is present, the cell swiftly responds by producing the necessary enzymes. So, in response to lactose, the Lac Operon switches on, producing enzymes to break down the sugar. This marvelous mechanism is a testament to the ingenuity of nature's design and a fundamental process in the world of molecular biology. But wait, there's another player in the game: glucose. Yes, that's right, glucose, the primary source of energy for most of our body's cells, also plays a significant role in the functioning of the Lac Operon. So, how does it do this? Well, it all comes down to the levels of glucose present in the cell. When glucose levels are high, the cell doesn't need to use lactose for energy. So, it makes sense that the Lac Operon, which is responsible for breaking down lactose, would be switched off, right? And that's exactly what happens. This phenomenon is known as catabolite repression. Catabolite repression is a form of gene regulation that allows the cell to prioritize the use of glucose over other sources of energy, such as lactose. This is because glucose is easier to break down and provides a faster source of energy. Here's how it works. When glucose levels are high, the concentration of a molecule called cyclic AMP, or CAMP, decreases. CAMP is a key player in the Lac Operon's regulation. It binds to a protein called the catabolite activator protein, or CAP. When CAMP levels are high, it binds to CAP, and this complex then binds to the Lac Operon, stimulating the transcription of the genes. However, when glucose levels are high, CAMP levels decrease, and this binding does not occur. As a result, the Lac Operon is not activated, and the genes for lactose metabolism are not transcribed. So you see, the Lac Operon is not just a simple on-off switch for lactose metabolism. It's a finely tuned system that takes into account the cell's energy needs and the availability of different energy sources. It ensures that the cell uses the most efficient energy source available, whether that's glucose or lactose. And that's not all. The Lac Operon also responds to changes in the environment, adjusting its activity to ensure the cell can survive and thrive, no matter what conditions it faces. So the Lac Operon balances not just lactose, but also glucose levels, showing its complex regulation. The Lac Operon, thus, is a master conductor of lactose metabolism in bacteria. It's an intriguing system of gene regulation, acting like a metabolic maestro, guiding the bacterial orchestra to respond to the presence or absence of lactose. This mechanism, though seemingly simple, has profound implications in the world of biology. The Lac Operon consists of 3 genes, the LAC-Z, LAC-Y, and LAC-A, each playing their own significant role in lactose breakdown. When lactose is present, the operon steps up, turning on these genes to produce the necessary enzymes for lactose digestion. It's like a sensitive switch, reacting to the slightest changes in the environment. But there's a twist in the tale. Glucose, the preferred energy source for bacteria, has a say in this process. When glucose is abundant, it suppresses the Lac Operon, even if lactose is present. It's a fascinating interaction, demonstrating how cells prioritize their energy sources. The Lac Operon's regulatory genes, the repressor and the promoter, are the unsung heroes. They regulate the operon, ensuring the right genes are expressed at the right time. They truly embody the essence of gene regulation. The Lac Operon, a simple system in bacteria, offers deep insights into how cells regulate their genes. It's a beautiful example of nature's intricate designs. So the next time you see a piece of cheese, remember, there's a fantastic microscopic concert happening right inside the bacteria that helps to break it down.